talked about for many years, scientists wrongly thought that proteins and not nucleic acids such as DNA were the inheritance molecule. So this question connects to why some people thought that way. So question 6 and 8 are going to connect, as well as question number 7 and 9 will connect in a moment. So for question number 6, how many different monomers or different types of amino acids could be found in polypeptides or small sections that would build proteins? So the answer would be there's 20 different types of amino acids that can be found inside of proteins in eukaryotic cells. Now, the next question is something that you may or may not have seen in your math classes based on what level you're in. This is from perms and coms, so permutations and commutations. So given the number of different amino acids, how many different combinations can form a sequence of exactly 3 amino acids? So we're going to make the assumption that in our 3 amino acid sequence, the same amino acid can be used more than once. So for the first amino acid in my sequence, it can be any of the 20 options. Assuming that the same amino acid can be side by side, there's also 20 options for amino acid number 2. And we make the same assumption, the third amino acid can be the same that appeared either in position 1 or 2, so that's also 20 different options. Our answer would be 20 cubed, or 20 times 20 times 20, which would give us 8,000 different combinations of 3 amino acids side by side. Now we're going to repeat the same type of calculation for DNA. So how many different monomers or nucleotides could be found in combinations in a DNA molecule? And specifically, when they say nucleotides, really they're talking about the bases in those nucleotides. So how many different types of bases have we seen in questions about DNA so far? I think Chargas rule from yesterday. 4. Adenine, thymine, guanine, and cytosine. So if we did the same perm and com calculation, how many different combinations of bases would be created if we have 3 nucleotides with bases side by side? So our first base could be any of the 4 options. Assuming that we can have the same base side by side, 4 options for the second base in my sequence. And then using our same assumption, the third base can be repeated. So also 4 options for base number 3. So we would have 4 times 4 times 4, or 4 cubed, which would be 64 combinations. So see how there's so many more combinations for forming a part of a protein than there is for a nucleic acid like DNA. This is part of the reason why people in molecular biology and also chemistry wrongly assumed that the protein might be the inheritance molecule early on, not DNA, because how could something so small and simplistic as DNA do such an important job within eukaryotic cells? And then the last question is going to be connecting to a little bit of some chemistry work that you may or may not have done based on your studies. So when cells are heated, the structures of proteins will warp and shift and denature. This means that the strands changes shape, and often they're unable to return to their original useful shape and no longer can do the function or job they're supposed to. When cells are heated, the structure of DNA will also warp, meaning the strands that make up DNA will break apart and the chemical bonds called hydrogen bonds that are holding the bases together will be broken. But the key difference between DNA and proteins is that when DNA strands are cooled again or the temperature is lowered, they will reanneal. So So our final question says, use the internet to discover what annealing means and what happens to the DNA strands when they re-anneal. So like the amoeba sisters say, let's go to the Google. Instead of doing that, I brought out one of our classroom models to demo this with you. So when we heat up DNA, the molecule will go from this normal spiral or helical shape and it will open to a linear or ladder shape. The added heat will help to break the hydrogen bonds between the molecules, and we would say this is now a warped or a structure that has become annealed. What does re-anneal mean? Literally the exact opposite. When the sample is cooled, the interaction will allow the hydrogen bonds to reform between the bases. When these chemical bonds, called hydrogen bonds, are reformed, that will allow the molecule to go back to its usual helical shape. So let's summarize that demo in words. So when we say that heating anneals DNA, it means the following. So I'm going to zoom in as much as I can. So upon heating, the hydrogen bonds, that is the abbreviation for between the nucleotides, are broken. As I demoed, this would allow the molecule to open up. And then re-anneal would be the opposite. So upon cooling, instead of breaking hydrogen bonds, the hydrogen bonds are able to reform. Between the nucleotides. And we would get a return to the normal or the helical shape of DNA. I'll give you a moment to get that written in. Did you say that shape called a helix? It's called a helix, yes. That's why you may have heard DNA is being trapped with a double helix molecule. Pardon? It's just specifically, it can affect other type of chemical bonding, but specifically hydrogen bonds is what helps to hold the sides of DNA together. And then for the last part of our class today, I wanted to talk to you and introduce you to the models about DNA that were being put together in the early 1950s, specifically between the years of 1950 through 1953. So for our note frame, this is going to connect to page 13 through the top of page 15. So we'll start talking about some of these important scientists. Their last names were Franklin, Wilkins, Crick, and Watson. And then tomorrow we'll be seeing almost 20-minute long video about determining DNA structure from the Howard Hughes Medical Institute, or HHMI, from the United States, which will give us more insight to what these individuals did. Okay, so by the end of the 1940s, biologists knew the following about DNA, a type of nucleic acid. They knew that nucleic acids were actually polymers, or molecules that were made up of the same unit that repeated itself over and over again. So we call these repeating units of nucleic acids nucleotides, and depending on the type of nucleic acid, DNA would have DNA nucleotides and RNA would have RNA nucleotides inside. So the next section with the bullet points would correspond to what is found in every nucleotide of DNA. So there would be a phosphate group, a 5-carbon sugar, which has the name of deoxyribose in DNA, hence the full name of deoxyribonucleic acid or DNA, and then 1 of 4 nitrogen-containing bases. So the 4 nitrogen bases that we would see inside of a nucleotide of a DNA molecule would be adenine, thymine, guanine, or cytosine, or A, T, G, or C for short. This is what the chemical structure of the base adenine would look like. And then if we were taking a look at RNA nucleotide inside, instead of thymine, we would see the base uracil instead. So it's kind of just a preview of what we're going to be getting into in more detail next week. And then I've included these diagrams for you halfway down page 13. So they were able to start to make sketches for what the appearance of the parts of nucleotides would look like. So this would be the physical, written, organic chemical structures, and then some people describe these as the ball and stick model to show what the atoms would look like in 3-dimensional space. So the hunt was on for how these nucleotides, these molecules, organize themselves into the structure of a DNA molecule. And they were trying to do this because they were trying to figure out what was the makeup or the 3-dimensional orientation of the parts that made up the inheritance molecule, which they discovered through the studies that we looked at yesterday was in fact DNA, not protein. So for discovering the structure of DNA, with the chemistry or the makeup of the parts of nucleotides known, it was up to chemists and other scientists to determine exactly how these parts of nucleic acids arrange themselves in 3-dimensional space and how they were able to store information that controls inheritance or the genetic information. If scientists understood this information, they were hoping that it would allow them to be able to somehow change or modify it and help them to understand how it made individuals different from one another. So in particular, there were 4 individuals whose work led to our development of DNA as being described as a double helix structure. And this was actually done through 2 pairs of scientists. So the first 2 on our list, a British chemist, Rosalind Franklin, and a British physicist and biologist, Maurice Wilkins, did some work together at the same college in England. And then the other 2 scientists, British physicist Francis Crick and American biologist James Watson, did more work at a different college in England. They both published similar thoughts about the structure of DNA in a very famous scientific journal called Nature in the same year, which was the spring of 1953. So we'll have a chance in a couple days to take a look at that actual journal article to see how it was presented to the scientific community. So we're going to, first of all, talk about the work of Rosalind Franklin. I'm going to say mostly her, because as we'll hear in the HHMY video, she was working with her colleague Maurice Wilkins, but remember 1950s females in the room was a much different time if you were involved in academia. And she did not get along with Maurice Wilkins because he thought that she was stepping on his toes and vice versa. So it was mostly Rosalind Franklin, but also Maurice Wilkins is brought into the discussion because they worked in the same department. So this takes us to the years 1951 and 1952. So Rosalind Franklin in particular made a single crystal of a DNA molecule, which at that time and still to this day is something very, very difficult to do in the world of chemistry. So she was able to take cells from the thymus gland of a calf or baby cow. She was able to extract the DNA out of those cells using the same procedure we talked about yesterday that Frederick Meisner created, and she was able to remove the liquid and create a crystal of just the DNA from those cells. She used a particular technique in her laboratory called X-ray crystallography. So this is a technique that's used to determine the atomic or molecular structure of a crystal in order to get an image of the pattern of how the X-rays bounce off of the crystallized sample. She was able to work backwards and figure out, based on what she saw in the pattern, what is the shape of the molecule that would give that particular pattern. So she initially wrongly, unfortunately, considered a triple helix based on what she was seeing in her photographs. This is a scan from her journal, and it says the next layer, 3 chain arrangements involve twigs, therefore a helix. So she looked at these photographs showing the pattern, and she wrongly thought there was a triple helix because this isn't what she thought it might be. She kind of put her research on the side and was focusing on other things. So specifically what X-ray diffraction does, this is just so you understand what her procedure was. She was shooting high energy X-rays at her crystallized DNA molecule. When the X-rays hit the atoms inside of the crystallized DNA, they would bounce off the atoms or diffract in different ways, and they would be picked up by a photographic film in her laboratory. Based on the pattern where the X-rays hit the film, she was able to deduce what the structure of the molecule would have to be to give the pattern that she was seeing. So when she did this in her laboratory, she collected 2 types of DNA patterns, and these are the pictures I gave you on the top of page number 14. So on the left-hand side is what we call the A form or the dry form of the crystallized sample, where water was removed to give a clearer or cleaner picture. So this is the one that she thought that would be 3 strands based on the pattern of diffracted X-rays she was seeing. This one is the critical one that was actually the most important part, but because it didn't match what she thought she was seeing in this one, she kind of left this one aside and put it away in her laboratory. We would call this the wet B form. This image is more blurry, but upon glance, do you see how it definitely has a characteristic X shape to it? It almost looks like a chromosome, but it really isn't. So this is the part that she was missing. She was so fixated on seeing a triple helix molecule, she may have been wrongly influenced by an American chemist named Linus Pauling, who thought that DNA should be a triple helix, that when she saw this, it didn't match what she or he thought. So she kind of left this one on the side, but actually this is what she should have been focusing on. This clearly shows a pattern that shows 2 banding patterns. Why would that be? Because DNA was later able to be discovered that is a 2-stranded helical model. So I'm going to just quickly slide this up so you can see the whole text. So the last bullet point reads, the plot thickens. So Marie Wilkins, her colleague she wasn't getting along with, shared this photograph called Photo 51, so the wet leaf bee-formed image. We call it Photo 51. I wish I had a great, awesome story to share with you, but it's literally the 51st picture. And when Wilkins' friends and colleagues, James Watson and Francis Crick, attended their university for a lecture, he showed them Rosalind Franklin's photograph without her knowing that he did so. This is what James Watson and Francis and Crick needed to see. It was their missing link in helping them confirm that DNA should not have 3 strands, but it should have 2 strands based on what they knew about X-ray diffraction patterns. So on the bottom, it says, to the naked eye, upon first glance, this photograph called Photo 51 kind of looks like an axe cut up into small bits or chunks, but it is what Watson and Crick needed to see to confirm that DNA was a double helix. So also around the time we're going to hear in the HHMI video that Watson and Crick did also attend a lecture that Rosalind Franklin talked about, but they didn't take any notes about what she was presenting, so they actually forgot about what she said. And then when Wilkins showed pictures, I was like, oh, the light bulb. Oh yeah, that's what she was talking about, and that gave them what they needed to do to create their model. So the missing gap on the bottom of page number 14, you may recognize this chemist's name. His name is Linus Pauling. Specifically in 1953, he proposed an incorrect model of DNA. He said that DNA should be a triple-stranded molecule where the bases of the nucleotides were on the outside of the molecule, not on the inside. If his name sounds familiar to you from chemistry, he was fundamental to the establishment of quantum chemistry, which is the entire first unit of the grade 12 chemistry curriculum here in Manitoba. He was also important in the establishment of creating models in molecular biology later in his career as well. So this is a photograph of his model, so you kind of get a computer sketch of that on the bottom of page 14 in the notes. So Linus Pauling wrongly thought that there were 3 strands of nucleotides that were intertwined to give the 3-dimensional structure of DNA, with the bases on the outside, not on the inside. One thing we'll never know for certain is there is a common legend in the history of science fields that some of Linus Pauling's graduate students recognized the error in his model chemically. The bonding between the phosphates and sugars would not work out due to this orientation. But because Linus Pauling was a little bit of a know-it-all, did not tell his advisor that he was wrong. So if we make a mistake at our school, please be great students and tell us along the way, please and thank you. So this takes us to the work of James Watson and Francis Crick in early 1953. So they were trying to put together an accurate 3-dimensional model of DNA. They didn't really do a lot of original work or experimentation on their own. Instead, they were analyzing pieces of information and data that were already proven by other scientists, but they were trying to look at this data and information in new and different or insightful ways. Now, this part I started to talk about a little bit. When their friend, Maurice Wilkins, showed James Watson and Francis and Crick the photo 51 Rosalind Franklin took, it's what they needed to see to give them definitive proof that DNA was not a triple helix model and instead should be a double helix model. With this part, you don't have to write down. This is just as an aside. Watson and Crick's first DNA model erroneously or incorrectly placed the bases on the outside of the molecule, just like Linus Pauling did. But based upon seeing this diffraction pattern in Rosalind Franklin's photo 51, they said, it should no longer be a triple helix model, and the bases should not be on the outside. It should be more like this. It should be a double-stranded molecule made up of nucleotides where the bases are on the inside, not on the outside. And then they also had to come up with a mechanism to propose how these bases were being held together, and that's where the concept of hydrogen bonding comes in. So to conclude our class, they said when they created their model called the double helix model of DNA in the spring of 1953, there were two sides of the helix that were connected by their bases. So we either had pairing between adenine and thymine bases or guanine or cytosine bases. And as a result, what they very literally did is they created metal nucleotides in their laboratories, and they were physically trying to connect them in space like puzzle pieces. So we'll see one of the presenters, James Watson, who developed the model, showing you what they did in the video tomorrow. This is how they put together their first accurate model of DNA, being described as a double helix. So once you get this written into our notes on the top of page 15, that will conclude our class today. So one final thing that I want you to do is do a quick double check in Google Classroom. Most of you have submitted your last assignment, but because part of class tomorrow will be doing a new assignment about the history of DNA we've been looking at this week, I want to make sure that everyone's caught up before we start doing a new assignment. So for some of you, that might literally mean press submit in Google Classroom. I saw some people were working, but they did not properly turn it in for So I'm going to stop recording at this point, but if there's anyone in the